Hi, I'm Alicia,
And I'm Samira.
and we'll be doing our project on invasive species and statistics. An invasive species can be any kind of living organism, amphibian, plant, insect, fish, fungus, bacteria, or even an organism's seeds or eggs that are not native to an ecosystem and cause harm. They can harm the environment, the economy, or even human health. Today, we'll be focusing on the Changing Hudson Project, which focuses on zebra mussel, or Dreissena polymorpha, which is a bivalve originally native to the Caspian Sea region. This species of mussel reached North America in the mid 1980s in the ballast water of a ship. They soon became established in the Great Lakes and the waters draining them. They will eventually colonize all the waters in North America except for those too warm or saline for their survival. Zebra mussels are filter eaters, meaning that they filter small organisms and organic particles out of the water at very high rates. This means that small edible material known as phytoplankton at the base of the aquatic food web are being filtered and strained out of the water at an alarming rate. These phytoplankton are what many native animals depend on for survival.
So this is just a visual to compare before and after the zebra mussel invasion. So the dotted line is an indication of the arrival of the zebra mussels in the Hudson River. And as you can see in figure B, the amount of zooplankton significantly dropped after the mussels were introduced to their environment. So for data collection, the population which we're deriving data from are the annual means for the density of zebra mussels and phytoplankton for the growing season, which is from May 1st to September 30th. The data were collected as part of a long-term study of the Hudson River ecosystem by researchers at the Institute of Eco Studies from 1991 to 2008. The populations living on hard bottoms were sampled by divers who collected 10 rocks at each of the seven sampling sites in June and then again in August. The populations were sampled using a stratified random sample design, and phytoplankton are sampled 4 to 6 times a year along the entire freshwater Hudson tidal river. As for our hypothesis, we hypothesized that the correlation between zebra mussel density and phytoplankton density is strong, linear, and negative.
The basic assumptions of the simple linear regression model are as follows. The distribution of E at any particular x value has a mean value of 0. The standard deviation of E is the same for any particular value of x. This standard deviation is denoted by sigma. The distribution of E at any particular x value is normal. And lastly, the random deviations of E1, E2, and so on are associated with different observations that are independent of one another. The scatter plot shows a linear pattern, and the variability in the phytoplankton, which is the Y value, appears to be similar across the range of the zebra mussel density, the X value. This supports the appropriateness of the simple linear regression model, so we assume the 4 basic assumptions are met. Doing our calculation, we find that the correlation coefficient is equal to negative 0.15. This is a weak negative linear association between zebra mussel density and zooplankton. The coefficient of determination is 0.023, which means 2.3% of the variation in zooplankton can be explained by mussel density. From the residual model, we can gather that the linear model is not appropriate since there is a clear curve in the residual plot and a pattern, and the points are not arranged randomly.
So because of this, we tried a quadratic regression model instead. So the quadratic regression model is definitely a better fit. We have a coefficient of determination of 0.505. Therefore, there is a moderate quadratic relationship between mussel density and zooplankton. So 50.5% of the variation can be explained by the appropriate quadratic relationship between mussel density and zooplankton. For our conclusion, as the model shows, our hypothesis was inaccurate. A quadratic regression model was a better fit for the relationship between zebra mussel density and zooplankton than a negative linear model. Approximately 50.5% of the observed variability in zooplankton is explained by the quadratic relationship with zebra mussel density. The magnitude of a typical sample deviation from the least squares line is about 326.545 zooplanktons per liter, which is typical deviation in zooplankton from predicted. So the question now really is, how much does invasive species abundance affect the environment?
Okay, let me answer that for you, Samira. Using what we have studied, our data from the Hudson ecosystem, the zebra mussel invasion has had profound effects on the Hudson River ecosystem. Phytoplankton and zooplankton are the base of the aquatic food web, and the zebra mussel's presence has severely harmed the other native species that depend on that food source for survival, and in turn set off the whole food web. The magnitude of these changes are comparable to disturbances in other aquatic ecosystems that are caused by toxins, nutrient pollution, and acid rain, to name a few. Like most invasive species, they were likely brought to the area by careless human activity. This is another graph that displays the ecological effects that have been brought on since the zebra mussels were introduced to the Hudson River ecosystem. As you can see, phytoplankton and zooplankton levels have decreased, and soluble P has increased, and other things that have changed the chemistry of the water. So why should you care? Well, let me tell you. Cost and impact to native species. It costs the US government $137 billion a year in damages and pest control costs. Zebra mussels threaten 49% of all endangered and threatened species. Furthermore, the mussel invasion is also responsible for 68% of all fish extinctions in the US. So think about that, America. Okay. Thank you for listening. Bye. Bye.